Now, when we go back to erythropoiesis, and we talked about this a little bit in the fetal erythropoiesis section of this chapter, but with erythropoiesis, we start with different forms of erythroblasts, normoblasts, right, orthoblasts, all of that, and eventually those are going to go on to form reticulocytes. Now, remember, with reticulocytes, there's a couple key things to remember. So, number one, the first thing is that these reticulocytes are essentially going to be going from the bone marrow to the blood, right? They're going to go from the bone marrow to the blood, and eventually in the blood, they'll eventually mature into erythrocytes, right? So in other words, we're going to have a fully matured erythrocyte. Now, the reticulocytes in general, in terms of their lifespan, is they're going to be about 1 to 2 days. Okay, so that's the next thing you want to remember, that reticulocytes in general have a lifespan of about 1 to 2 days, whereas erythrocytes, especially in adult, will typically have a lifespan of 100 to 120 days. And remember, the consequence of this is that when you're looking at a reticulocyte count in general, the reticulocyte count is going to be quite low, usually around 1%. And like I said, we discussed this all in the fetal erythropoiesis section, but the reason for that is partly because of the lifespan, right? The reticulocytes are going to be far short-lived relative to your erythrocytes in general. Okay, and so we see that here with the lifespan. Now, what is really controlling and regulating this process? Well, this is all regulated through erythropoietin. Okay, so remember, erythropoietin is released primarily from the peritubular cells of the kidney, specifically the peritubular fibroblast cells, but we're just going to put kidney here. And again, you want to juxtapose things like this, right? So thrombopoietin, we said, is going to be synthesized in the liver and the kidney. If you have liver dysfunction, you can see platelet dysfunction, right? Specifically quantitative platelet dysfunctions like thrombocytopenia, for example, if a patient has liver disease. Similarly, right, if a patient has kidney disease, you might expect these patients to have low red blood cell counts, right? In these situations, we could say that that patient might have an anemia. Okay, they might have an anemia secondary to kidney dysfunction, right? If I have kidney dysfunction, that would predispose me. You might be saying, well, why does that predispose you to, an anemia? Well, the reason is if the kidney's not working, the kidney's not able to release erythropoietin to stimulate erythropoiesis, to stimulate this process to eventually form red blood cells. Okay, so that's the concept. Remember, erythropoietin is stimulating the bone marrow primarily, right? Because that's where these reticulocytes are coming from. So the erythropoietin is going to go to stimulate from the kidney, it's going to go into the blood, and it's going to go to stimulate the bone marrow because that's where those red blood cells are coming from. Now, it's going to stimulate erythropoiesis specifically through the JAK2 STAT pathway. Okay, and you might be saying, this is kind of a lot of detail. Do I really need to know this? Well, the reason this comes up is if you understand the basics here, then it's going to be pretty straightforward when you run into diseases like polycythemia. Okay, so for example, polycythemia here is going to be very classic, specifically polycythemia vera is going to be very classic for having mutations in this JAK2 STAT pathway. So you can imagine if you had a mutation in this pathway, specifically if you had a mutation that causes that pathway to get activated, right? In those situations, you might expect these patients might be getting a lot of red blood cell, a lot of erythropoiesis if we had an activating mutation there potentially. And we'll talk about that in a lot more detail when we talk about polycythemia, but this is just putting those, basic concepts together. What would it look like mechanistically if a patient has an anemia, right? Kidney disease being one thing, or what would it look like potentially if a patient has polycythemia? Well, it might be an activating mutation in that JAK2 STAT pathway that causes overactivation, that causes us to produce too many red blood cells, okay? And that's essentially, and again, that's what polycythemia is. It's an overabundance of these red blood cells. All right, let's talk about the structure a little bit of these red blood cells. So, generally we're talking about an oval biconcave disc, and it's difficult to, kind of get lost with the red blood cells from some of the other cells, okay? So you can see these are all red blood cells here, a little bit of central pallor, and you can see here in this electron micrograph, you have this biconcave shape to the red blood cell. And then you can see here, this is actually a platelet, right? We talked about this a little bit in the last video. And then this structure here, this is going to be a granulocyte. So this is what they look like, under an electron microscope, just to give you an idea, a three-dimensional idea of what we're talking about. In terms of the structure, again, the other big thing that you want to know, and this is particularly high yield because it has a lot of repercussions when we talk about some other things. Erythrocytes don't have a nucleus, and we talked about this in the fetal erythropoiesis section. They really don't have, most organelles either. So, if you're trying to differentiate, for example, a reticulocyte from an erythrocyte, one of the things that we talked about is that the reticulocytes will have cytoplasmic RNA. Erythrocytes in general won't have that. And like I said, they'll have a completely absent nucleus. Remember, as you're transitioning through this process, one of the things that you'll notice specifically with the erythroblasts is that eventually the nuclei will start to kind of shrink, and eventually we're going to lose that nucleus entirely. And the whole purpose behind this is so that we can maximize the surface to volume ratio of our red blood cells so that we get a more efficient oxygen transportation. Okay, that's the concept. And the hemoglobin that's going to be transporting that oxygen is going to be within the cytoplasm. And again, we're trying to maximize the space for that. And with that being said, that hemoglobin, like we just said, is going to be transporting oxygen primarily. The other big function of the red blood cells is that we're also going to be transporting CO2, right? Carbon dioxide. So we're inhaling oxygen in the lungs. We're going to bind that oxygen to red blood cells that will take that oxygen to our tissues. Those tissues will be giving off CO2 when you talk about glycolysis, TCA cycle, electron transport chain, right? We're going to have carbon dioxide released from those tissues through metabolism, and that CO2 is essentially going to be exchanged in red blood cells using the enzyme carbonic anhydrase. And, I don't want to get too far off track, but essentially that enzyme in conjunction with your chloride, bicarb, antiporter. So these two things are largely going to drive the carbon dioxide transport. And we'll talk, there's a lot of this discussed in the pulmonology section. Big picture here, right? We're just looking big picture. The red blood cells are essentially going to be transporting oxygen using hemoglobin, transporting CO2 through the use of carbonic anhydrase, and then using this chloride bicarb antiporter so that we can get the bicarb out of the red blood cells at the lungs, right? And then we can get that bicarb to then form CO2 and we can exhale it out of our lungs. And again, I think we talked a little bit about the pathology. Again, anemia in general, we're talking about, a low hematocrit, low red blood cell count, polycythemia, where we have an elevated count. And again, another term you might see for polycythemia, sometimes you'll see erythrocytosis. Okay, so you might also see this written as erythrocytosis, kind of like thrombocytosis when we talked about platelets. Okay, so again, these are going to be more quantitative, right? So we're talking about, a low or a high level of our red blood cells, but you can also have qualitative problems. You can have problems with the structure of the red blood cells. And so some of these problems might include things like anisocytosis. Anisocytosis specifically is referring to changes in the size of the red blood cell. So you might have a wider spectrum of sizes with anisocytosis. Poikilocytosis is referring to changes in shape. And sometimes you'll have a combination of these. It just depends on the disorder. There's things like hereditary spherocytosis, where we have a more spheroid or elliptical shape to the red blood cells. And again, we're going to talk about all of these. Again, this is just an introduction, but it's important to remember, when we're talking about platelets, when we're talking about red blood cells, we kind of categorize these into quantitative and qualitative defects. And one thing that I want to remind you of, just because we'll talk about the type 1 hypersensitivities and their relevance to basophils and mast cells. But just remember too, when we talk about certain types of anemias, for specifically autoimmune hemolytic anemia, or if we were talking about platelets in the earlier part of this chapter, we can have situations like immune thrombocytopenic purpura, where we have antibodies that are targeted against either the red blood cell or the platelet or both. Okay, and that red blood cell being targeted can then have an antibody binding and then complement-mediated destruction. And we discuss all of this in the immunology unit, but this is just a reminder that things like autoimmune hemolytic anemia, which is what this stands for, autoimmune hemolytic anemia, where we essentially have antibodies binding to the red blood cells and then basically marking them for destruction using complement. That can also result in an anemia, right? And that's through type 2 hypersensitivity reactions. And again, we'll go through all the specifics on this. It's just giving you an example. Again, this is big picture. In terms of the relevant pharmacology here, it's also important to remember that EPO can be given synthetically, EPOetin being probably the big one. And again, these are synthetic forms of this hormone. The important thing to remember, first of all, when would you even use these? Well, remember, patients that have kidney disease, chronic kidney disease, usually will have very poor erythropoietin release. And the reason for that is because they're released from the peritubular cells of the kidney. So if the kidney is not working, it's not releasing EPO. We already said that, right? So you might have very low levels of EPO in these patients, and so you might give them, some synthetic analog. The thing about this is when you start to give patients synthetic erythropoietin, you're going to stimulate the bone marrow to produce red blood cells at a much faster rate. So sometimes what happens here is you start producing red blood cells so quickly that your iron stores get completely depleted, because the iron stores are essentially trying to keep up with this rapid proliferation of your red blood cells from this synthetic EPO. So in a lot of these patients that have a normocytic anemia from their kidney disease, they can actually develop a microcytic iron deficient anemia when they get synthetic EPO. And we'll discuss these different variants when we go through the anemia section. That's more of a advanced concept, but just something to keep in mind.